Don't worry, we are just getting started. If you guys are joining online, we haven't done anything in Kahoot or anything, so you haven't missed anything with that. There is a video posted in the discussions for week 11. The week 11 review video is just a video of me going over the actual immune response. So if you guys haven't watched that, I recommend that you do that. Okay, Kahoot. And then we're gonna that login. I don't know why I asked so many times about the cookies.
I didn't know what cookies were until like a week ago.
That's all right. Usually don't worry about it too much. Okay, Kahoot. Let's do chapter 17. No, we haven't done 17 yet. I guess we could do 16 again. Let's just do a couple questions from 16. If I can find it. 15. Oh, okay, thank you. This one. Post live. Let's get everybody signed in. Start. Okay, there we go. I had a student, so while you guys are getting signed in, I had a student in my night class who we did a 1 question Kahoot for attendance, and she was late for that 1 question. You know, we start class in this class, it's supposed to start at 10 a.m. and I was running a little late today. It's already 10:12, talking about some of this stuff. By the time that we got that 1 question sign in and everybody was done with it, that student apparently arrived into my class at 13. Oh my God, now I've got to do math in my head. No, it wasn't even 13, it was even 10 minutes. She was 8 minutes late to the class, okay? The reason that we have the Kahoot in the beginning, your attendance points for the class are not based on whether you show up to class at all. It's based on your participation in the Kahoot, and that's stated in the syllabus. And so if you miss the Kahoot, you miss the points. It doesn't matter if you show up later. And that's just how it's stated in the syllabus, and that's how I run my class. So if you wanted your points, you have to show up on time. That's to get everybody here to participate on time. If you don't show up on time, you don't get to hear me say things like, be sure you're watching that video. You don't get to hear me say things like, hey, make sure you do this, that, or the other, and you don't get to participate in the Kahoot. You might miss that stuff. So I want to make that clear. Even if it's just a 1 question Kahoot, if you're late for it, you're not getting the points for attending it. You're supposed to be here on time, right? And if I'm running 2 minutes late and we start the Kahoot late and you still miss that, you were late. Like, that's just how that goes. So if you're later than even me, you're later than me. So that's how that goes. I also want to add, I don't have an issue with me being here more than 15 minutes late. But if you've had professors that show up more than 15 minutes late to their class, 15 minutes is the cutoff where you are allowed to say that that professor is not showing up and not coming to class. So if you ever have a professor that shows up more than 15 minutes late to their class and you leave and they try to give you points off or count you for being not in attendance or something like that, that's against school policy. The university policy, not just that professor can say or do whatever they want. Doesn't mean they're going to get fired. I want to be clear on that, right? A lot of these people that have tenure, it takes a lot to fire a tenured professor. I mean, a lot to fire a tenured professor. But having said that, you can't be held accountable after that 15 minutes, okay? Because you haven't had a professor that's been 15 minutes late yet. You probably will. It's within your rights after 15 minutes to say that professor isn't showing up and you don't have the say. And they can't hold you accountable for having missed that content either. That's on them at that point, okay? FYI with the late thing. I show up 5 minutes late, that doesn't count as far as that stuff goes. It's my responsibility to be here on time, but 15 minutes is the cutoff for that stuff. Just to point out that we're not even at 15 minutes right now. Okay. I just happen to know there are professors that have showed up more than 15 minutes late to their classes before, and I just want you guys to be aware of your rights in those things. So we're going to get started with this Kahoot. We're going to do 2 questions, just because that's what I can afford the time for. All right, where do all lymphocytes begin development? We've done these questions before, but now we have a better perspective on it. All lymphocytes begin their development in the same place. That is and again, we're not going to be doing the Kahoots for 17 or 18 in class. I'm not even going to do them on Wednesday, okay, before our presentations. When our Wednesday's our presentation day if you're on a Unit 3 presentation. So we're not going to do those Kahoots, but they are available on Canvas if you want to use them for practice or go over those questions on your own time. All of these Kahoots are. All righty, next question. During maturation, lymphocytes weed out anti-self clones and will only allow appropriate clones to proliferate. The term for that whole process. And we did go over that stuff in chapter 16. All righty, so that is going to be clonal selection. So here's our little scoreboard. I'm going to end it here. Okay, we're going to end this here. And we're going to jump into chapter 17. Why does this say 1? Why does this say this? Team chat. I don't even know what that is. Okay, I'll look at that later. All right, so chapter 17. Let's give our overview for this chapter. Chapter 17 deals with disorders in immunity. So immunopathology is the term that refers to a disease state associated with the immune system. So it's usually from overreaction or underreaction. If you're overreacting, we call that hypersensitivity. If you're underreacting, it's a hyposensitivity. We also call it immunodeficiency. We're going to talk about our hyposensitivities at the end of the lecture. So when you're underreacting, your immunodeficiencies, that's going to be at the end of the lecture. First, we're going to start off with our 4 types of hypersensitivities. Again, they're called just type 1, 2, 3, and 4. But at the very, very general big picture, when we're talking about these guys, it's important to understand type 1, what type of immunity is associated with that, and et cetera. Okay, and I'll make a little chart up on the board as we go through this stuff, just to keep us on track. We'll have our hyper. And we're not going to have class questions today. I thought that's kind of obvious though, because I don't want to take the time for it. I will review the questions though when we get to them. As for hypersensitivities, we have type 1, 2, 3, and 4. They're usually written by Roman numerals, but that's not as important. And then we have our hypo, which are our immunodeficiencies. And there are 2 types of these. There's just 2, and it's either primary or secondary. And we're going to talk about them and what causes each of them as we go through these, right? So hypersensitivity are 4 major categories of type 1, 2, 3, and 4. These are going to make more sense, our little explanations up here when we get to them. But type 1, this is led by IgE. Type 2 is either IgM or IgG. Type 3, we're really talking about complexes like agglutination, basically, with IgG. And then type 4, we're going to be talking about cell-mediated. Which, if you don't know, cell-mediated immunity is driven by your T cells. Okay, so all of these 1, 2, and 3, they're all antibodies. Type 4 is going to be your T cells. And when we get into primary and secondary, we'll talk about what those are. They're just a little more straightforward. So let's get into our type 1s that are led by our IgE response. Normally, IgE is going to be associated with attacking our parasites. Did it talk about it on this? It did not. It's probably in the next slide. It's associated with normally attacking parasites. But when our body isn't trained earlier on in our life on how to use IgE to attack parasites, that's your protozoa and your parasitic worms, and your body misuses that and basically overacts to things that are not parasites or protozoa using those same mechanisms. That's where allergies come from. And essentially, this is going to be hypersensitivity. Basically, having any immune response to something that's not a pathogen, we can consider that a hypersensitivity. You're being too sensitive to something that you really don't need to be reacting to at all. That's our hypersensitivity reaction. So these are our allergies. And allergens are anything that can elicit that sort of inappropriate response. We have two types of allergy severities, atopy and then anaphylaxis. So atopic allergies are going to be local. Even when you have eczema that affects the skin, it doesn't affect your internal organs. So eczema is local, it's the skin, it's not your internal organs. But if it's spread systemically, that's when we see anaphylaxis. And that response is also systemic. And so that's when we start having fatal reaction, usually associated with a shock response, all triggered by that IgE reaction. All right, so here's the hygiene hypothesis. This is to say, I saw one student say, if we were exposed to dirt, we wouldn't have allergies. It's not that simple, okay? So the hygiene hypothesis says that because of our hygienic environment, we're not being exposed to as many of the antigens that we would be if we weren't as hygienic. And that is to say, underdeveloped countries, like third world, what we call third world countries that don't have these strict hygiene things as part of their society, they don't have anywhere near the incidence of allergies. And so it is hypothesized, it is proposed that that may be due to the fact that they are getting exposed to antigens like protozoa and helminths that are helping train their immune response to behave more appropriately. That's the hygiene hypothesis. Not that being exposed to dirt makes you not have allergies. We could break it down because there's another thing that we hear about that deals with first world countries, which is you might live in a first world country, but if you grew up on a farm, you're less likely to have allergies. Now, that's not because you got helminth infections, but it's because you're exposed to more antigens. It's a less clean environment. You are dirtier. So there is an association with that, but it's just not as simple as that, right? And so that goes up to more like dealing with how much exposure you have to all the possible antigens in the world. The cleaner you are, the less antigens you're exposed to. And then that can lead to inappropriate training of your cells. If you want to think about it as dirty equals less allergies, that's okay. But I feel like understanding the more complex side of that, especially when we're talking about societal differences, it just helps with a better understanding of how the world works. Common allergens can be exposed to your body by inhaling, ingesting, injecting, and contacting directly, like with touch. And these things we have some crossover in some of these guys as well. Inhaling something, you could be inhaled pollen or dust or animal dander, things like that, but you could also come into contact with some of these things too. So there can be some crossover with injectants, bees, like bee or wasp. I'll make sure that this is, yeah, it's still picking up, okay. If you are having exposure to these sorts of things, you may not have ever seen them in your body before, and so you never got trained that they're something you shouldn't react to. If you're not able to produce it, you never got trained that it is something is self or something you shouldn't react to. So there's issues with those sorts of things, but if it's getting injected, you have a much higher probability of having an anaphylactic response to it. Can you have an anaphylactic response to something that you ingest? Of course. And we know this with peanuts. If you're allergic to peanuts, you can have an anaphylactic response from like it being in the air, from actual inhaling it, from ingesting it, or from contacting it. But you can have an anaphylactic response because once your body sees it, it thinks, oh, my whole body is probably getting attacked by this thing, and you have a systemic response to it. And so some of these things Yes, question.
Allergies, a lot of people can't, like when you cook something, you can't cook it on top of something that you're allergic to beforehand,
Mhm.
like gluten allergies, you know?
Yeah.
Is it because it's not like alive, so it doesn't kill under the heat?
Right, and so it's, yeah, it's not gonna be gotten rid of with like gluten or peanuts or anything like that. Even by denaturing that a little bit with the heat, what you're allergic to might not get denatured enough. It might still be in present enough form, but the heat isn't enough to break it down and turn it into something you're not allergic to anymore. Yeah. And so we see that with certain proteins, and that the fact that they are carried over even after being exposed to heat suggests that the heat isn't denaturing it enough to change the shape for your antibodies, not to know the difference. That's a great question. And so we're kind of aware of which category some of these things would fall into. So let's walk through how that allergic response happens. So your allergen acts as an antigen here. So we're seeing it as foreign and reacting to it as though it's foreign. So the allergen gets in, and your B cell sees it, and it gets activated by your T helper cell, and it's able to, proliferate and differentiate. And the plasma cell, the effector cell of the B cell, makes antibody against this thing. For whatever reason, there are signals that are sent with this allergen that say we need an IgE response. And so it triggers switching from IgM. Instead of switching from IgM and favoring IgG, it switches from IgM and favors IgE because of signals that are associated with how this allergen was introduced into your body. So now you've got the allergen, you're making IgE against it. IgE is grabbed onto by mast cells. So you remember the different granulocytes we talked about with the fills and mast cells. Mast cells have like basically a flagpole holder on the surface of their cells. And that flagpole holder grabs on to that constant region of IgE, that Y-shaped antibody. They're still shaped the same, but the IgE gets grabbed onto by the flagpole holder on the mast cells. And the mast cells basically wear those flagpoles are going to be like, they're going to grab onto the constant region of those antibodies, and the variable regions that interact with the antigen are on the outside. And so if those antibodies, so it wears it like a receptor. It creates a new receptor for itself out of the IgE that it's wearing around. And when IgE sees antigen, it causes your mast cell to degranulate. And that means releasing all of its inflammatory signals. And that includes histamine and other things like that. So what's actually happening whenever you're allergic to cats, like I am, or whatever you may be allergic to, pollen or whatever, that your body is seeing this antigen, and it is triggering your mast cells that are wearing the IgE to degranulate. And those mast cells can wear that IgE against pet dander or whatever for decades. And it'll just stay in your body just waiting to see pet dander again, and then it will degranulate and release histamine and all these things. And that's why antihistamines are so effective at controlling allergy, because it's basically blocking those chemicals that are being released by your mast cells from causing the inflammatory reaction to occur. All right. So that's what's happening at that actual cellular level with those allergies. Those IgE molecules prime the mast cells to react to that allergen, and then the mast cells release their inflammatory mediators as a result every time it sees it. And so this leads to things like hay fever and asthma. Again, these are atopic. These are local reactions. So hay fever, allergic rhinitis. When you get an itchy, sinuses, watery nose, and itchy eyes from these things, that's still a local reaction in your upper respiratory area. Allergic rhinitis. Allergic because it's associated with an allergic response. Rhinitis, rhino means nose. We kind of know this already with rhinoplasties and things like that, like a nose job. Or itis always means inflammation. Anytime you see itis at the end of anything, like meningitis, that's inflammation. In meningitis, itis, inflammation of the meninges. And encephalitis, itis, inflammation of encephala, which is the brain itself. And so anytime you see itis, it always means inflammation of the part that precedes it. Okay, so rhinitis, inflammation in the nose. And that's all that's happening with hay fever. You can have rhinitis, not from allergies, right? You can get inflammation in your nose from having a cold, for example. That's not allergic rhinitis. That's going to be an actual illness from actual pathogen, but it's still rhinitis. Okay, so with allergic rhinitis, You're having an IgE response to whatever you inhaled in your nose for that. With asthma, this is a local allergic response in your like actual respiratory system, your lower respiratory system. And so you're having IgE response in your actual lungs. The inflammation will associate there, and then the medications that we take as steroids that you inhale help abate the inflammation response there, but it's still local. Eczema is atopic dermatitis. So we have atopic dermatitis, which is allergy on the skin, derma, skin, itis, inflammation, and it's atopic because it's local to the skin, and it's associated with IgE. We will talk about another, like we've talked about, what was it, poison ivy earlier, right? We talked about that in the last chapter. Poison ivy is this one down here. You're still having an inappropriate reaction to the oils of poison ivy, but contact dermatitis is a type 4. Atopic dermatitis is a type 1. And it's different. Okay. Moving on, food allergies from ingesting things or even breathing in the particulate from the food or something like that, or contacting the particulate. But a lot of times if you're getting it from ingestion, this can lead to GI symptoms, but not always. As it turns out, I have an allergy to whey protein of all things. It's not super sensitive, but if I have too much whey protein, I have severe GI symptoms, including pancreatitis and things like that, associated with my GI system reacting to whey protein. Does that mean I'm going to cut out cheese? I'm sorry, but no, there's no level of me having a cheese allergy. I'm actually allergic to the whey, but I just have it in moderation. And so I have to be aware of how much how much basically inflammation I'm willing to put up with from my allergy. But some people have food allergies that trigger anaphylaxis. And it just depends entirely on how your body as an individual is reacting. When it comes to your immune system, you really can't compare it to how other people's immune systems react. Because it's very individual to how your body reassorted your genes. And so I might have a whey allergy and nobody else in my family has a whey allergy. My genes just reassorted that way. Luckily, they reassorted in a way that I only get GI symptoms and not anaphylactic shock when I ingest these things. And so that difference can be, what we see. So if you're ingesting something, you might still have other reactions in other areas of your body as these allergens are spread throughout the body from exposure. Drug allergies can range anywhere from a mild rash to anaphylaxis, and it also depends on how it's delivered to the body, whether it's from contact or injecting or from oral ingestion of drugs. That can have a difference on whether or not you're triggering a rash or anaphylaxis. Anaphylaxis is characterized by respiratory and circulatory disruption. It can be fatal within minutes. And I want to make this very clear. If you have an allergen that could potentially trigger anaphylaxis and you use an EpiPen to treat it, epinephrine, also known as adrenaline, was delivered by the medical community. We usually call it epinephrine. If it's produced by your body, we call it adrenaline. But epinephrine is only going to reverse the effects of that inflammatory response, but it doesn't get rid of the antigen. So epinephrine can treat you for 30 minutes to an hour, but once that epinephrine wears off, if that allergen is still present, you're still going to trigger anaphylaxis. So if you only carry one EpiPen, that is the time that you get bought that gets you to the hospital. If you have an anaphylactic response and you've had to use an EpiPen, or you know somebody that's had to use an EpiPen, they should be going to the ER, not just being, oh, I feel better and going on with their day, because that allergy can still be there to trigger anaphylaxis. This is actually extremely important because once that epinephrine wears off and isn't, forcing your body to bring blood flow, forcing your body to bring in air and things like this, that epinephrine wears off within 30 minutes or so. And now suddenly you're having an anaphylactic shock and you don't have another EpiPen. What do? You die. That's what you do. And probably by that time, if you're in a place where you can't drive yourself to the hospital, if you haven't called 911 after your first use of your first EpiPen use, you aren't going to have time to wait for the ambulance to get there after that first EpiPen wears off and you realize, oh, you've made a mistake. The EpiPen does not cure or treat the actual anaphylactic thing. It only treats the inflammatory response that's going on in that exact moment. So it's not going to prevent it from coming back, right? And we also know if it treated or cured the thing, then one EpiPen use would cure you of your allergy. That's not how that works. It only treats the symptoms of what is going on at the time. So if you know anybody, or if you use an EpiPen and you've had to use it, or they have had to use it, or you have a patient that had to use it, they should be seeking medical attention immediately after they realize that they're having a potential anaphylactic reaction. Not use it and they're fine after that. If they used it and they're fine after that, they may have removed themselves from the allergen and they might be okay. But if they used it and they're not sure where the allergen came from, or they haven't been able to remove themselves from it, like if it was an injection or if it was a bee sting or something like that, they need to be getting emergency medical attention immediately. Because this can happen within minutes. And if you know anybody that has an anaphylactic response to something like this, and you probably know this, that if they receive a bee sting or if they're exposed to something that could cause anaphylactic, like a shellfish allergy or something, they will know and start getting symptoms within minutes of the exposure. And they usually have to use the EpiPen right away before it gets to a point where their breathing is constricted or their blood flow is constricted to their brain. Because what the body thinks is going on is that you've been exposed to this massive amount of pathogen. And it doesn't want a pathogen getting to the brain. It doesn't want the pathogen being able to get into your body through your lungs. And so it closes those things off. And it thinks it's just going to be temporary. It doesn't know it could potentially kill you by doing that. The body doesn't know that. It's a response that's in place, meant to be to protect you from parasites and protozoa and things like that. So if you had a parasite or protozoa in your blood, this could potentially be a helpful mechanism, but would also be regulated completely differently than how it is with our allergy responses, right? So anti-allergy medications will either inhibit IgE production or inhibit the inflammatory mediators that come after exposure. So like histamines, bradykinin, and things like that. Steroids that just inhibit the immune response in general. Steroids are great for inhibiting your immune response, especially if you're having like allergies or if you have a pathogen that causes itis in other areas of your body, whether it's your throat, pharyngitis, or your nose, rhinitis, or sinusitis. When you have those inflammation responses going on, steroids are great for reducing that inflammation. But if you keep it up long term, the reason steroids are good at reducing inflammation is because they inhibit your immune response. So if you keep it up for too long, you're going to be more susceptible to infections. So you can't use steroids long term. That's actually why doctors often will be a little hesitant to prescribe steroids, because they don't want you to become dependent on that for controlling your inflammation, which could lead to effects later on down the line. Anything that you take as a drug to inhibit your inflammatory response will increase your likelihood for infection. I think we're going to talk about one in a little bit, but anyways, we can also interfere with the histamine response itself. And that's how antihistamines will work, is just by blocking histamine from being able to attach to its receptors. Some of our histamine responses can also affect other aspects of how your body functions, and some of them can cross the blood-brain barrier. Zyrtec, Claritin, Allegra, things like that, those are antihistamines that cannot cross the blood-brain barrier. So they don't make you quite as drowsy. Benadryl, extremely effective at inhibiting histamine, can cross the blood-brain barrier and affect your actual brain function. Well, that's why you feel so sedated when you take Benadryl. That's why it makes you tired. But it's very good at inhibiting histamine, and it's very quick at doing it as well. So epinephrine is what we use to increase our airflow and increase our blood flow to our brain, but more than one dose can be required, and most people only carry one EpiPen, so it's why you need to get to the hospital if you have an anaphylactic reaction. We can actually desensitize you to your allergens, to some of them. And this has to be tailored to each allergen. So let's say you have a pet allergy. I can inject you with small amounts of cat dander allergen. It's actually, I don't know if you guys know this, but in cat allergies, you're not allergic to their dander, you're allergic to the saliva that they clean their bodies by licking themselves. And then the saliva is attached to like all of their cells, all of their hairs, and it gets everywhere and whatever. It's a molecule in the saliva. But anyways, the antigen, cat saliva or whatever, we can inject you with small amounts of it, along with chemokines or cytokines of cell signals that like trigger certain kinds of immune responses. So cell signals dictated that we should make IgE in the beginning. But if I put that antigen in your body and put signals to make IgG, we already know that IgG, if you are able to make IgG, makes up 80% of all of the antibody that you'll make. IgE is like 0.02%. But if you favored this response and you didn't get IgG, then your allergen will bind to IgE. But if you make IgG instead to that allergen, IgG will bind up with all of that allergen and IgE will never see it. IgE is your workhorse, I'll make more of it, it binds stronger. So IgG can actually eliminate the allergen and prevent IgE from ever seeing it. So it can trigger your body into making IgG that'll clear out the allergen and prevent IgE from ever seeing it, even after you've had the allergen. And so this takes a lot of injections basically acting as vaccines over time to basically induce your body to switch to IgG production to block the IgE. And this is a successful way of treating allergens in cases where we've been able to develop this for each allergen. It's not available for every allergen. But maybe in the future, you know, that's how that will work. So now we're going to move on to type 2. So IgE response, this is going to be basically allergies, atopic and anaphylactic. Okay, so IgE type 2, this is going to be our blood antigens. Okay, so type 2 reactions is basically you have antibodies, IgM or IgG, normal antibody response against an antigen. Because I have type A, type B looks foreign to me. Okay, so to me, because I have type A, my body's never seen type B, and my body will treat B as an antigen. So it makes IgM or IgG to attack this antigen that's foreign to my body. So if you give me blood that is from somebody that is B, like my dad is a B positive and I'm an A positive. If I were to get my dad's blood, my blood would have antibodies against his blood and would attack it. So you might think, okay, so it gets rid of his blood. That's fine. Once you start killing somebody's blood cells and breaking them open, your body gets this response and signal saying, what the heck, blood cells are getting broken open. There's something going wrong in my body. And that leads to shock and death within minutes. So you cannot give somebody the wrong blood type. You're infusing them with antigen, and their body will attack that antigen and think that we have in our bloodstream antigen, not blood, but antigen. And so my body attacks it, and we get a shock response as a result of that. There are 4 blood types, A, B, AB, and O. Okay? You can have genes from your parents as follows. If you have an A or a B, they are equally dominant. O is recessive. That means if you have an A gene, you will express A. If you have a B gene, you will express B. If you have an A and a B gene, you will express both. And O is basically the foundation on which all of them are built. So think of it as you have a foundation of O, and you might have A sticking off of O, or you might have B sticking off of O. Everybody has O as the foundation. That's actually why everybody sees O as friendly. And everybody can receive O. Regardless of your blood type, I'm an A. Regardless of the fact that I'm A, I can receive O because O is also my foundation for the A. It's always friendly to everybody. Everybody has that as their foundation. Yes, question.
Like in the pharmacy I work at, people like MS and stuff will kind of get infusions, which is IgG infusions. And those are like blood derivative products, right? So it's the person who gives that only ever type O, like, instead like the blood and that not react with whatever.
So when you get immunoglobulin infusions, it doesn't have blood cells. Right. So these are markers on your actual red blood cells, not on any other cell in your body, only on your red blood cells. The red blood cells have A or B or O, or some combination of those. And that your other cells, your white blood cells and things like that, when you have immune globulin, they take the plasma out of someone, and the plasma, which is the liquid portion of your blood, it has the proteins like antibodies in it, and that includes your immune globulins. And so you can infuse somebody with those antibodies that way, but it doesn't have the red blood cells, and it usually doesn't have the white blood cells either. So that's what will be happening there. Okay, so everybody has O as their foundation. And then it's whether or not you express A or B or neither on top of that O, okay? Now, if you have a parent that is an O blood type, I wish I don't have my screen marker. Let me try to draw it. Oh my gosh, okay, pen. If you have a parent, like my dad is a B, and my mom is, I'm just gonna say she must be an A, I don't know, I wish I could undo that, but whatever. My mom must be some kind of an A. So if this is my dad, oh my God. That's a D, by the way, that's a D. And if this is my mom, and I am an A. Well, if you have to get a gene from your mom and a gene from your dad in every single gene that you have in your entire body, you got one version from your mother and one version from your father, always, in every single gene in your body. If I had to get one from my mom and one from my dad, and I don't have B, and I just told you, if you have B, you express B. If you have A, you express A. If you have both, you express both, okay? I can tell you automatically that my dad's genetic makeup is that he must have one B and one O, because I didn't get a B. So if he had two B's to donate, I would have had to have gotten one of the B's. He must have had a B and an O. So my actual genetic makeup must be A and O. But O is recessive. And if you have an A, you have to express it. If you have a B, you have to express it. But if you have an O, you have to have two O's. You can't have an A or a B in order to just be O. Okay? Yes.
So if somebody were to have A positive and then the other O negative, would the children most likely have A? Or
No.
would it-
No. Okay, I'm gonna tell you why. Okay, so this comes back to something that we don't learn in this class, which is called a Punnett square. So if somebody has, like me, I know my genetic type is AO. I has to be because of my parents. And this promise when I tell you that I know that they're my parents, okay? So genetically speaking, in order for my dad to be a B positive and for me to be an A positive, he had two genes. One of them was a B and one of them was an O. He was expressing the B, so he's a B blood type. The O is there just as the foundation. And so the O is there, and so I must be an AO. If I have a partner that is B and they're carrying an O, just like my dad was, okay, here's the possibilities of what all of our offspring could be. Wow, I just really messed that up. Sorry, give me just a second. Eraser, erase this line, pen, okay. Okay, so here's our square, all right? This is our square. Now we could have a baby that would carry the A and the B, or we could have a baby that carries the A and the O. This is a no, I promise, okay? Or we could have a baby that carries the B and the O. Or we could have a baby that has an O and an O. And just like that, you have a parent that could just be expressing A and a parent that is just expressing B, but they could have a baby that could have any of the four blood types. So they could have a baby that's AB, they could have a baby that's A, they could have a baby that's B, or they could have a baby that's O. This is probably the genetic makeup of my parents. My mom might be an AA. She might have carried two A's in her alleles. I don't know. If that were the case, if we had an AA, if it was AA and BO, this were my mom and my dad. I don't know, because you don't know what their other side is unless you can look at the babies. But if it was AB as a possibility here, and then we have A, I don't know why it does that thing where it like shoots off like that. It's not something weird I'm doing. Or we could have an AO or an AO. This combination of genetic parents cannot have a baby with just an O or a baby with just a B. You see? So I don't know which one my mom was. I don't know if she's AA or AO, but I know that I'm an A positive and my sister's an AB positive. That's all I know. All right. Are you guys following a little bit with this? Okay, so that's the genetic side of this. So the four possible blood types that can result from any of these genetic combinations are either A, B, AB, or just O. If you're O, then your genetic makeup is OO. That's your genetic makeup. That's it. You are not, like A can be AA or A can be AO. But if you have A on there, you're expressing A. End of story. Okay, so let's look at this. This is what it looks like at the foundation. This is type O, and you see how all blood types have this going on as their foundation. It's a common marker for everybody. And then it's just whether or not you have A or B or both added on to that. And if you don't get any genes for A or B, then you are not going to express A or B. You'll just have the foundation. Okay? Okay. If you are A, then B looks foreign to you. If you are B, then A looks foreign to you. If you are AB, neither of them look foreign to you. And if you are O, A looks foreign to you and B looks foreign to you. So everybody can receive O because O, everybody has it as their foundation. O is the universal donor. But if you're AB, A doesn't look foreign to you. Can you get A? Yeah. B doesn't look foreign to you. Can you get B? Yeah. You're AB, so obviously you can get AB. And O, everybody has it as a foundation. So can you get O? Yeah. So if you are AB, you can receive any blood type. That's the universal recipient. If you are O, you are the universal donor. But people with the O blood type can only get what kind of blood? O. Okay? As it turns out, you would think that based on these combinations and all these different reassortments and everything, that would associate with the frequency of those genes in the population, but it doesn't. The most common blood type in the United States is O positive. It's recessive, but it's the most common blood type in the United States. As it turns out, O comes along with other benefits. First of all, if you are an O blood type, you smell better to mosquitoes.
I know.
Okay, hate to break it to you. My husband's an O positive, and let me tell you, I can assure you that that is a true thing. All right, because if he comes outside with me, I don't get any mosquitoes. He gets all of them. And so O smells better to mosquitoes, but O is also more resistant to malaria. Having the A and the B makes you more susceptible to malaria infection. And so O is favored in the human population over a long period of time, well before, we had society and things like we have now, first world countries, that O was a favored genetic trait to have. And people with O are less likely to have malaria or have it as seriously. And so they would survive better and they passed on their genes more. So that's why we think O is more common. And mosquitoes have evolved to favor O, so they can try to make up for that difference and try to infect more of the O people and try to still have their survivability. Don't we all love a mosquito? But anyways, just some tidbits of information on there. All right, we talked about our universal donors and recipients. If I transfuse you with the wrong blood type, it activates complement against you. You have antibody that's going to be made against the blood type you can't receive. If you're AB, congratulations, this isn't ever going to be a problem. But I'm A, so if somebody transfuses me with B, my complement will be activated by the classical complement pathway, and then we'll have those membrane attack complex holes getting poked in all the red blood cells, and it just causes them to break open and lyse. They release chemicals that tell your body that you've got some serious issue going on, destroying blood cells, and that leads to shock and fever and inflammation disseminated throughout the body and can lead to death within minutes to hours. And so this can, if you survive having the wrong blood transfused, it can cause permanent damage as well. But during that process, in order to save somebody that's getting the wrong blood type, we'll have to immediately start dialysis to start filtering out all those toxins produced by damaging those red blood cells. Their spleen could develop splenomegaly from trying to deal with the damaged red blood cells. And I mean, managing all those symptoms all at once has to be something that you are ready and prepared to deal with immediately. You don't have time. If you start realizing you're transfusing your patient with the wrong blood type, you don't have time to call in from the pharmacy for drugs to help do all this. You don't have time to call in to have a dialysis machine moved in and start hooking that person up with a central line or something like that. Usually, you don't have time to manage those things. And so that can lead to death very quickly. Death is a common outcome of an incorrect blood type. So we don't just have A and B and O, right? We also have the Rh factor. And so this is whether you're O positive or O negative, or A positive or A negative. That positive and negative on there is whether or not you have this factor called the rhesus factor, Rh. I won't test you on rhesus, but you do need to know that on your blood, there are two possible antigens, the ABO blood group and Rh group, okay? And it's just whether you have it or not. If you express Rh, you are Rh positive. And this is a dominant trait as well. So if you have just one gene of positive, you'll be positive, okay? And if you have two genes of negative, then you'll be negative. So if you have a gene from mom that's positive and a gene from dad that's negative, you'll be positive. You have to have two genes of negative to be negative. All right, so if you are negative for RH, you see positive. If somebody has RH, you see that as foreign. This is a problem when you are pregnant. In hemolytic disease of the newborn, this is why we give Rogam. Rogam is an antibody that blocks antibodies to RH. So if you are RH negative and you have a baby in your belly that is RH positive, your body sees the RH as foreign and develops antibodies against the baby in their blood. This doesn't happen until later on in the pregnancy, okay? And it takes a while for that immunity to develop. And it isn't usually going to be activated until we have an issue with that placental barrier getting broken during birth. And so that's when the antibodies can actually start acting. So late in the pregnancy, you start having this reaction to the RH, but it doesn't show up in your first pregnancy. That's your sensitization step. Just like your immunizations against a disease is to sensitize you to that thing so that your second response can be instant. The next RH positive baby that RH negative mother had, she will attack it faster. We usually don't see the deadly effects until birth is happening because we have disruption of that placental barrier. So you can carry a baby to full term, and then whenever you give birth to it, it'd be stillborn. It's actually extremely common in situations where you have an RH negative mother having their second RH positive baby. If you have carried a baby to a certain point in pregnancy and then miscarried it later on, you could still have this sensitization. And this is why, even if you've never had a baby previously, we will treat you with Rogam if you are RH negative. Especially if we don't know the blood type of the father, and especially if we know that the father is a positive, we're for sure gonna give Rogam. That's gonna prevent the antibodies of the mother from attacking the baby. This can save a pregnancy. Trust me when I tell you, this is not something that happened in the past olden days. This is something that still happens today. Imagine you are a mother, and you're either not aware of your blood type or your blood type of the father, or you are switching doctors that kept track of that stuff for you. You go to a new doctor, that doctor doesn't have that sort of knowledge of your blood type, and you didn't think that needed to be shared or something, and now you're going and seeing a new doctor, and they don't know anything about your past history. People still today give birth to stillborn babies because of this. And I will tell you, I have at least every semester had at least one person that had direct experience with this. Imagine carrying your baby for 9 months and then it being stillborn because of this, a preventable thing. That's why Rogam is pushed so hard for our RH negative mothers. Nope. Yeah, it sucks that it's that expensive. It should be part of what we would consider universal health care, but we're not going to get into that discussion, right? So if you want to talk about insurance and how it's a scam, we can get into that some other time because I'm with you. Yeah.
So basically, there's no way to tell if you don't know blood types. It'll just happen.
Yeah.
Like you don't feel it.
And so did you have something to say, Gavin?
I did.
Okay. So I was going to say in modern medicine, this is why we will do blood tests on the mother. And if you are RH negative, we're just going to assume that there's going to be an issue with this and push Rogam, whether we think it's your first pregnancy or your second pregnancy. We're not going to care because we're going to assume you may have potentially miscarried and had a reaction to this before. We're just going to assume it. And so this is something that we push so heavily with that if you are RH negative, you test RH negative here in the United States, especially, we're going to give you Rogam. And it's going to be one of those things we're considering life-threatening to the baby. Yes, did you have something to say to that?
Is this can be as RH positive?
Okay, so if you are RH positive, you don't see RH negative as foreign. RH negative just means you don't have RH, right? And so if you are an RH positive mother, you don't attack an RH negative baby. Well, you might start thinking about, okay, then if the mother sees RH as foreign, wouldn't an A mother see B baby as foreign? And yes, that's true. So my mother is A, we've already established that. And my sister is AB. So she had B in her body, right? Turns out that this reaction with the RH is significantly stronger than with the ABO groups. You can still have hemolytic disease of the newborn from the ABO differences, but that's when you're going to have your bilirubin babies that need to be under the bili lights. The baby is born yellow and you have to put it under a light. Probably the mother was a different blood type than the baby. and attacks the baby, but it's not as severe with those differences. Also, you have to have pre-sensitization. So if my mom is A and she had her first baby was A, which is me, and her second baby had B, which is AB, but there was B there, she was never pre-sensitized to that. So my baby sister probably didn't have any bilirubin issues. There wasn't pre-sensitization. Okay, are we following with that? All right. All right, next is our type 3. These are our immune globulin IgG complexes. If you make IgG against strep throat, and that IgG is floating around in your body and complexing with strep throat pathogen parts, antigen, right? That it'll create antigen to antibody to antigen, and they'll like agglutinate with each other. And that's an immune complex. When we have antibody and antigen cross-reacting with one another and creating these complexes, they can get caught in your basement membrane, which is like basically the membrane at the top of your tissues, okay? This isn't so much of a big deal in our larger vessels that can sustain some damage and not have an issue, but in our thin capillaries where we only have one layer of endothelial cells, this inflammation that can be triggered by antigen and antibody being there can lead to overreaction. So this is actually what we see with lupus. In lupus, we have antigen and antibody complexing together and getting stuck in the capillaries, like in the face. And we see the butterfly rash from that. You guys know about this with lupus. Yeah, they get that red rash on their face. These are antigen antibody complexes getting stuck in those capillaries. And the capillaries essentially are getting ruptured by the inflammatory response. This is a big deal in the kidney, where you have lots and lots of tiny little capillaries that are filtering things out of the blood and into the kidney tissue and cycling it into the ureters for filtration. So if you have strep, Streptococcus pyogenes is group A strep. If you have a reaction to strep, the antibodies can complex with the antigens and get caught in your kidneys and cause damage to your kidneys. This is a symptom called acute post-streptococcal glomerulonephritis. I will not make you write it down, but you better be able to recognize it on a test. Okay? Acute post-streptococcal. It's acute because it's due to this particular incident. It's not going to be chronic. It's not going to last for a lifetime. But while the antigen is there and antibody is binding with it during that acute phase, reacting to Streptococcus pyogenes, which is strep throat, group A strep, you can get glomerulonephritis. The glomerulus is a structure in your kidney. Nephro means kidney. Itis means inflammation. All right, moving on. Type 4 hypersensitivity is essentially very similar to what we have going on here. It's antigen not binding to antibody this time, but to the cytotoxic T cells. So those cytotoxic T cells that relate to your inside antigens are reacting to things that are not pathogens. So like poison ivy, it thinks that poison ivy is something inside of your cells, and it attacks them that way. When you have reactivation of antibody, that response is instant. We've talked about that. Your memory B cells can just start cranking at that antibody right away. With your T cell response, your memory cytotoxic T cells can't just start killing right away. They have to first, your memory cells proliferate out, and then those proliferated memory cells can start killing. It takes 2 to 3 days for those cells to proliferate. This is why poison ivy doesn't show up until 2 to 3 days after you see it. Or when you wear earrings that make you get that weepy, if you know, man, you know. Well, I'm allergic to metals. And so when I wear certain kinds of metals, I'll get this weepy, crusty, and my ears get really inflamed and stuff. It's this, but I get it 2 to 3 days after I wear the earrings. Okay? You have to wait on that. Another example of this is whenever you get your tuberculosis skin test, the PPD tuberculin skin test. We inject tuberculosis antigen just under the skin. 2 to 3 days later, we see if you have a swelling. Why do we wait 2 to 3 days? We've got to wait for your T cells to react. If you have a T cell memory response, that will react within 2 to 3 days and swell up. It takes that long for those cells to get reactivated. If your cells are reactivated within 2 to 3 days, congratulations, you've seen tuberculosis before. You either have it or you had a vaccine against it. Those are the reasons for why you would have a positive skin test to tuberculin. The only other reason is because you had a weird cross reaction to some other antigen in there. And so then if they're not sure, if you have a positive skin test to tuberculosis, do they just say you have tuberculosis? No. And if you haven't had the vaccine, do they just say, well, the only other way would be if you had it? No, they do a chest x-ray. They can do a chest x-ray and it shows damage in the lungs. you have, congratulations, tuberculosis. And believe me when I tell you, you can carry it around without any reactions. Contact dermatitis, not atopic dermatitis, which was eczema. Contact dermatitis is this, type 4. It's like poison ivy and other haptens, like metals and jewelry and things like that. And then that happens because the T cell response is going and killing your skin cells and causing this inflammatory reaction from killing your own cells. This is also the basis for graft rejection. Your MHCs that are in your cells right now, your MHC1s, are just sitting there, not presenting anything bad. They're just chilling. They're just MHC1s. They would be presenting inside antigen, but they're not doing anything when I get your kidney. But my body sees that as MHC is here, but it doesn't look right. And so it attacks it with this. It looks like it's not self, but it looks like something's coming from inside because it's still MHC1, but something's wrong, so we kill it. And it takes this. So this is when we get graft rejection. You can have the obvious thing, like I get your kidney and my body attacks it. That is the host attacking the graft. That's the obvious one. But if you get lymphocytes from a bone marrow donation, the lymphocytes from the bone marrow, if it's not matched, they will attack the body. And that is graft versus host. When those lymphocytes come from somebody else that didn't match you, they'll see all of your whole body as foreign and start attacking it. That's graft versus host. Running out of time. These are the 4 classes of grafts that I'm going to touch on briefly. Autograft is from yourself. Isograft is from somebody who is directly genetically identical, but identical twin is the only option for this. Or if we have cloning in the future, your clone, okay? And then allografts are from other people who are not genetically identical to you. If I get a graft from my younger sister, there's no way that my younger sister is genetically identical to me, right? So that would be an allograft. That's not an isograft. They have to be genetically identical for isograft, right? Then we have xenografts that come from other species. So when you get a heart valve from a pig, which is pretty common, that's a xenograft. Autoimmunity is whenever these things start leading to attacking our own tissues. And there are some examples of some autoimmune diseases and which type of hypersensitivity led to those diseases. We think that some of this arises from molecular mimicry, where the molecules on the bad guys look like self molecules, and then we accidentally start attacking self molecules as a result. And we think that things like this, like strep, we think that strep has molecules on its surfaces that can look like self. That's where we get rheumatic fever from. We also have just infection in general, potentially causing activation of other cells at the time because we don't have proper regulation of that immune response. So it can also trigger anti-self reactions. We think this is how MS develops and possibly type 1 diabetes. That infection has accidentally caused, or reaction to an antigen has accidentally caused the rest of the immune system to be activated. And then that has led to us attacking the cells for those things. So attacking the cells in your pancreas that make insulin, which is what you get in type 1 diabetes, or literally an immune response attacking the cells that make insulin and destroying them permanently. Or MS, attacking the myelin sheath, the coating on your nerves, leading to inflammation in the nervous system. And that could be triggered by viral infections, we think potentially relating to measles and things like that, but we're not entirely sure yet. We start seeing more indication of measles, we might see more MS. Yet another reason to be a proponent for the measles vaccine. Okay. The gut biome is actually really important for training our body and making sure that we are attacking things that are attacking things that are not self and not attacking things that should be friendly, and in which areas of the body. You can have all these microbes in your gut, but if they get into your bloodstream, that's a whole other story. And so these guys are actually really important on training your body on how to react to things. Finally, in our hypo or immunodeficiencies, we have primary. These are from genetic defects that are present from birth. Or secondary, these are acquired. Let me tell you, acquired immunodeficiency syndrome. What is that? AIDS, right? AIDS, acquired immunodeficiency. So what type is it? Secondary. Okay. So HIV infection can lead to the development of AIDS. We know that that happens because it attacks your CD4 positive helper cells. Those are the captains that are important for activating your B and your T cell response. And so if you can't have any B or T cell response, you have a very high susceptibility to infection. That's an immunodeficiency. We're not reacting enough, and it is acquired because of HIV infection. All right. This can affect your B cells or your T cells. Obviously, if we're affecting T cells, it's going to be a bigger deal because that can also affect your B cells, generally speaking. All right. A gamma globulinemia is people who cannot make antibody because their B cell response is messed up. Their T cells, though, can otherwise function normally. You'll have some increased incidence of infection, but it won't be anything like what we have with our T cell problems. If you have a T cell problem, you can't govern either side. We usually see a bigger issue. DeGeorge syndrome is an example of that. All right, severe combined immunodeficiency, like SCID, this is like boy in a bubble. This is where we have actual dysfunction on both sides. So your B cell response and your T cell response crack all together. And so if you have no B cell response and no T cell response, this is the most severe version. X-linked SCID is an example of this. So X-linked skin, the reason why it's boy in a bubble and not girl in a bubble, because X-linked skin is because men only get one X chromosome. So if they have issues with genes that are on that X chromosome, they don't have another chromosome to compensate for that. And that happens with X-linked skin. There is a gene on the X chromosome that is responsible for your entire acquired immune system. So these people cannot have any memory ever for any of the infections they see. They can just get reinfected with everything. All right, so anything that's acquired later on in life, either from infection or because you had radiation therapy for treating your cancer and that's depleted your immune system, or you have cancer that affects your immune system, like with now I can't think, lymphoma, that's the word, or leukemia. These are cancers that affect your immune system. And so when you have those types of things, you might have an immunodeficiency that's acquired because of getting the cancer. All right, but if it's present from birth, that's primary. If you got it later in life, that's secondary. It could be from infection, like with HIV, or from cancers that affect the immune system. All right. Really quick, I want to touch on the things from chapter 18. I'm not going to have time to go over these questions. So I just want to jump into chapter 18. I didn't even pull it up originally, so bear with me. This is what I meant to click on. Lectures, unit 3. Chapter, where is it? This isn't even right. Is it? Let it be 18. Here it is. Okay, so chapter 18, my basics that I want to get off to you guys before we go. There are 3 categories, just 3. Well, there's kind of 4. There's another category, but it's a different thing. There are 3 major categories for our diagnostics: phenotypic, immunologic, and genetic. Please understand the following. If it involves interacting with an antigen, it's immunologic. If it relies on recognizing an antigen, it's immunologic. If it relies on DNA or RNA, if it relies on nucleic acid sequence, is genotypic. Everything else, basically, is phenotypic. So if we're looking at your morphology, so actual colony morphology, your cell shape, whether or not you were gram-positive, whether or not you grew on this media or that media, or what this means for your actual metabolic breakdown, those are all phenotypic. They don't involve antigen, and they don't involve genetic sequence. If it involves antigen, it's immunologic. If it involves genetic sequence, it's genotypic. If it involves any of the other things, stain results, whether it grows on media, or that media growth tells you what is going on physiologically or metabolically or whatever, that's all phenotypic. So some phenotypic tests that we haven't discussed previously. So some of these will require culturing, and some of these you can test directly, right? And then based on what tests are positive or negative, we can say if you're positive for the gram stain, okay, if you're positive for the gram stain, did you have endospores or not? Did you react to catalase or not? Did you do this or that? And if you did or didn't, we'll go down different branches on our dichotomous key, which is what we're going to be doing for the lab at the end of the lab. Once we have all of our results, if you're positive for gram positive, and then whether or not you were cocci or bacilli, and then if you're cocci, did you react when we put hydrogen peroxide, if you have catalase or not, and then whether or not you grew a mannitol salt auger or not after those steps. You always start from the top and work your way over. You don't start in the middle and say, oh, my thing grew on mannitol cell agar. It must be this. Okay. All right, you have to start from the top. We'll go over that in the lab when we actually do it. Biochemical testing, whether or not the microbe has an enzyme or not, based on if it grows on this media or causes this color change in the media. We can do these tests like we're doing in lab. There are these cards where you can put your culture in, and it tells you the answer to all of these tests all at once. They're also extremely expensive. We're talking like $1,000 a card here. I can't afford that for all of my students. So we're doing the tests individually the slow way, okay? But these are really expensive. And if we are not sure at all what bacteria is causing your disease, we can put it on this, and that'll give us an idea of, oh, this is probably an E. coli based on this. And then we can do a genetic test to confirm that, okay? Can't just shotgun genetic tests, so we can do that in mass. And we can also use Kirby-Bauer. If you are resistant to this antibiotic, that might give us an indication that, oh, Streptococcus is always resistant to this antibiotic, and that can help with identification with those things as well. Phage typing. Every single bacteriophage that ever existed only infects one bacterial species. So if I think you have salmonella, and I'm like, okay, I have salmonella phages here, and I put it in the sample, and it causes your bacteria to not be able to grow because the phages infected your bacteria and burst them open, and now they're all dead and they couldn't grow, then you probably have salmonella. However, I put salmonella phages and your sample still grows, it could be E. coli. So that's what phage typing is. A particular species of phage only infects a particular species of bacteria. And so we can use that specificity to identify things. All right, cultures, whether it grows or not, or whether it causes color changes and things like that, all phenotypic. The problem is some of this relies on culturing, so it could be delayed 24 hours or more. And if things are very slow to grow, that might not be an option. So phenotypic diagnostic methods, looking at it directly, the cell shape, how it stains, culturing it, and then looking at selective or differential growth, biochemical testing, whether it can use this or that, or can ferment this or that, susceptibility testing, and then things like phage typing. Immunologic methods is also known as serology. It depends on antigen recognition. It's usually dependent on antibody as well, just like ELISA. ELISA is an immunologic method. These can be direct or indirect, and serology usually uses serum, as is in the name, but it can use other things as well. We can also find antibodies or antigens that are present in other specimens as well. So if we're doing direct or indirect, we remember direct asks the question, does the patient have the antigen? Indirect asks the question, does the patient have the antibody, right? And so we're coming back to that. This does not just apply to ELISA. It applies to all immunologic tests. All immunologic tests. Does it have the antigen or does it have the antibody? So it's direct or indirect based on that. This can be agglutination, causing things to agglutinate and visibly be able to cause like something coagulating in the bottom of the well. It could be precipitation, which is a smaller complex of just antibody and antigen, not whole pathogens. So we're using whole cells, that's agglutination. If we're precipitating just antigens and antibodies, that's precipitation. This could be immunochromatography, linking colors to things, so we can actually see color differences in the cells. This is very similar to ELISA, where we have color changing wells. But immunochromatography, we're going to be looking at individual cells, not just a whole well changing in color. We can label cells with colors that way too. And this could be using fluorescent antibodies that have color, or it could be like immunochromatography where we have like your home COVID tests. Clearly your home COVID test causes a color change in a test, but is it in wells like we see with ELISA? No, so it's just immunochromatography. If it's in wells, it's ELISA, and if we're using fluorescent antibodies, then it's immunofluorescence, okay? So that just depends on how you're looking at it and which category it falls into. Western blot, I'm 99% sure Western blot is on the test, okay? Western blot is a system where we are running the proteins through a gel, just like DNA. So you can push DNA to the positive end of the gel. You can also push proteins to the positive end of the gel. Run it through electric current, through a gel. They separate based on size, the exact same way as the DNA did, okay? So that process isn't different, but now we're going to use antibody to see if the band is there or not. So this is running something out on a gel, putting it onto a special piece of paper, and then on the paper, seeing if the antibody binds there or not. If it does, then the antigen is there. That's how Western blot works. And Western blot is extremely important for verifying HIV. So what might do ELISA might say this person has antibody to HIV, but then Western blot to confirm that. It's more specific and more sensitive than ELISA. In vivo tests are tests that take place inside of a living body, like our tuberculin tests. That's a test to see if you have antibodies, right? Or if you have, it's actually T cells. So it's a test to see if you have had a T cell response to this antigen, and that's an immunologic test that takes place in a living body. So that's in vivo. In vitro means it's in a test tube. In utero is in the womb. Okay, so that's the antibody stuff. And then really quick, again, there's more information on these things for when you guys are studying this stuff. Then the genetic testing relies on the actual nucleic acid sequence. This could be by using PCR to amplify the sequence. If it amplifies, you're positive. If it doesn't amplify, you're not. For hybridization, we are using a specific genetic sequence and seeing if it matches up with the thing that's there. We can do this in situ. This is not in vivo and it's not in vitro. What in situ means is I can take a biopsy of your tissue, and on the actual tissue that's still intact, see if my probes bind with that. We can use fluorescent DNA labeled with fluorescent markers. If that fluorescent marker sticks to the tissue, it means you have the genes for that, mutation or something like that. And we talked about this previously, remember, in chapter 6 and 8. We talked about these methods. And so this is just putting this into perspective that this is a genotypic method. So that's FISH. We've talked about FISH before. Yeah. And we can sequence a whole genome as well to identify things and can make comparisons. And we already talked about gel electrophoresis. But fluorescent labeling of DNA markers, in order to see that the DNA marker binds to the cells, indicates that the genes for whatever you're looking for are there. Be fluorescent carrying that with it. We can also put a microarray. Again, we have introduced this before in chapter 8. A whole big old card with all these different genetic sequences on it, mash up the patient cells, mix it in with that, and wherever we see binding, that person has those individual genetic markers. That can be used for identifying different types of cancer as well as different microbes. And we can actually use all these technologies and put them on these little microchips, and we can take a blood sample from a patient out in like a third world country and test them for all these things with these high level, very sensitive, very specific things. It's just expensive to get all these tests out there to these areas, but it could make healthcare more easily usable for places like third world countries. MALDI-TOF is mass spectrometry. It's bombarding a sample, like a patient sample, grow up the culture, put it into the mass spec, and then shooting basically lasers at it. And then the parts that break off ionize and float in the air. And then we measure how long it takes them to hit the sensor. That's called time of flight. And that's what MALDI-TOF time of flight does. It basically measures how long the different parts of the molecule take to get to a sensor. And based on that can tell you the exact molecular makeup of something. And that can tell you exactly what it is. It's not used for everything, but it's very helpful in identifying drugs. We can also use imaging. These are obvious things, right? We all know how MRI, CT scans, and X-rays can be used to image patients to look for things as well. And that is it. Let's head up to the lab and get started. Thanks for sticking with it, guys. You did great.